So thanks, Ryan, for letting me know about this workshop, and thanks everyone for letting me talk at this. So I'll keep it fairly short because Raf showed a lot of things that we did, and Raf, Ryan, Nicholas, and a lot of other people were involved in getting me up and running with all of this work that I'm going to show. So essentially, I work on this technique called high energy diffraction microscopy. We can study different types of materials. We can study jet engine turbine blades. We can study electronic circuit solder joints. We can study radioactive materials. We can study granular materials such as sand. And the idea there is that we want to be able to look inside materials without destroying them. And then we want to be able to do that relatively fast. So originally what we had been doing was running this workflow at our local cluster. And now we are, with the help of FuncX, Gladiator portals, and Globus Automate, and the whole Globus infrastructure, we are moving towards a workflow which looks something like this. So let me actually bring the whole slide up. Okay, so the way it kind of looks like, the way we are trying to implement it right now is something like this. So we have three techniques: this far field HEDM, this near field HEDM, and tomography. And the idea is that we have these different types of detector which save some data locally on an intermediate storage. And this data is then using Globus transfer copied onto a long-term storage. And the thing that we are trying to do is using Globus transfer to copy the data over to ALCF, so some remote machine, because even ALCF is remote for us because we are not sitting at the machine itself. And then using FuncX, do all the analysis for all these three modalities, do some merging to get different information about the material, put everything together, and then do some simulation. And then the final goal, as Raf also showed, is to kind of try to drive the experiment using these results. So it becomes very important to be able to not only just do the analysis, but also do some visualization, get some real-time feedback of the experiment. Now, the way the current FuncX flow that I wrote, that we developed looks like is something like this. So as soon as a complete data set is generated, this workflow kicks in. And the workflow basically kicking in means that we have a few steps in the workflow. So the first step is inject. It takes the data from Voyager, copies it over to Theta for processing, and then all of these steps run on Theta. So we have some pre-processing step on a single node, and then these two steps here, so this image processing and indexing refinement steps, these run on multiple nodes, and then these three steps in between, they run on single nodes. And then at the end, we have this extract and publish step, which takes the data, copies it back to Voyager, and also it publishes to this Gladiator portal, so that people can look at how their results look like. Now, to kind of show how the flow itself looks like, it's only maybe 5-6 lines of code, and those 5-6 lines say, okay, set up the input, set up the payloads, and now set up the flow, and then go ahead and run the flow, right? And then you get the results. Of course, it's not as simple as that. I think everyone would be familiar here with what it is inside. So using these Gladiator tools, it's very easy to set up a flow. So this is how we define this flow. So we say, these are the tools that we are going to use for defining the flow, and each one of these is a single step, right? So we have this inject, remote prepare, remote peak search. So this is multi-node, remote transforms, remote index refine, and so on and so on. So we have these different steps. And now if I want to run the same flow on a different modality, so on a single node, I have a different flow that I have defined here. I say run these in this order, and then essentially changing the flow is as simple as changing this command from this to something else, and it will run that file that we have, right, that we have set up. Now, how does one of these files look like? It's actually relatively simple. So I just import some things, and I call the C code that I have written from Python and run it, right? So that's as simple as that. Now, I think one of the nice things that we had to develop that we struggled a little bit was how to tell the FuncX engine that these are different nodes and how to provide them with different arguments. And one nice trick that Nick Saint came up with was to define two functions. So we have an argument builder function, and we have the real function. And basically, what we tell it here is that this main function requires the tasks. The tasks to be provided to this main function are the output of this argument builder function, right? So, and that was kind of neat. I was surprised how well this worked. And this args builder function is defined here, and basically what it says is in the data that it got, the multiple tasks unwrap this and set up this nice payload to be provided for different tasks of this peak search function. And now what does this multiple tasks have? So let me go here. So what this multiple tasks has is a number of parameters, but what is important for us is that this block number it should be different for different instances, right? So for each node, this number should be a little bit different, and this is how we set it up. And doing all of this, running this flow, things are very neat. We are able to ingest onto the portal and see the results as soon as we run the analysis. So this was in very short time what we are doing with FuncX and Globus and Gladiator portals and Globus Automate.
Very cool. So I have one quick question before anybody else jumps in. I know you had this set up to run on various different clusters. So you were able to run your flow targeted at Theta or targeted at Orthros, the APS cluster, or Cooley at ALCF. Were you doing anything around packaging up your tool, or were you just installing it in the same location so the same function could be used in different places?
Yeah, so this is something that we want to look into next, is using containers, right? So right now we are installing things on different places, and we are a user facility. So I have about 25 users who run my software on 25 different machines all over the world. And right now what it does is it uses a version of SwiftK that I originally developed. And what we are planning to do is sooner rather than later, trying to move away from that and use the new FuncX flows. And then, yeah, using the containers would help things a lot instead of installing things for each machine from beginning.
Cool. Any other questions?
I have a question. So I was curious, I saw that you had indexing as part of your flow. I'm wondering, is that done via like just a software script that you just send all the way down to the files, or is it, or do you have that? I know you mentioned you're not using containers yet, or do you have like some container that maybe you're using at this point to, run a whole bunch of indexing tasks?
Oh, sorry. So this indexing actually means taking the diffraction data and grouping it into different grains.
Okay.
So this is not the indexing in computer science terminology. This is more indexing for us to group the different diffraction peaks into grains. So and we have a code which does that.
Okay, so maybe what I was thinking of then was more in your extract and publish step then.
Yeah.
Okay. Very cool.